Welcome back to Office Hours Unplugged, where we chat with the most prominent researchers here in McMaster. Today, we finally get to talk to Dr. Layla Soleymani. Thank you so much for joining us today, Professor. Could you please introduce yourself to the audience?
Sure, yeah. I'm Layla Soleymani. I'm a professor in Department of Engineering Physics, and my research is focused on biological sensors, and I'm thrilled to be here.
Awesome. And then just kind of take us back to how your journey through academia kind of started.
How far do you want me to go back?
As far as you want.
as far as
yeah.
Okay, good. I guess in school, like in high school, I was kind of the kind of person who loved all sciences and didn't really know what to specialize in. I think that's something that a lot of people feel. And I like chemistry, I like biology, and I like physics. So because I came from a family of engineers, I was encouraged to just go for engineering, and then you'll figure out what to do next. And so that's kind of where I started.
Right.
And so I did a degree in electrical engineering at McGill, and I was exposed to a wide range of topics. Even though it's electrical engineering, it was an interesting time. It was kind of the start of the bio-nano era.
Right.
Like right now, everything is about AI. At the time, everything was about kind of bio-nano. And it was perfect for me because I didn't know really where I wanted to specialize in. But the bio-nano kind of gave me an opportunity to be a multidisciplinary researcher. It kind of opened that door because in nano, you could be a chemist, you could be a physicist, you could be an engineer, a biologist, and there's a space for everybody to contribute to the advancement of science. So this is where I got into this bio-nano, and I knew that that's where I wanted to kind of to stay. So then I again, as I said, I didn't really know where to specialize, but I said, okay, I'll do a master's and learn more about this bio-nano world. And so I went to the United States. I went to University of Southern California, and the program was in a way that I could explore different labs and different projects. Right. And I really got into at the time, like working with these nanomaterials and exploring their application, particularly in biology. And so this is, it kind of solidified my path that, I wanted to kind of stay in this space, the nanobio, and be in a multidisciplinary environment, which has kind of guided my whole career. Right. And then I got really interested in the work of this professor at University of Toronto who worked at kind of this bio-nano interface, and I decided to come back and do a PhD. And so this is where I started to know I'm going to build an academic career. Up until then, I was exploring, and I'm glad that I had those opportunities to explore. And then I had quite a few options. Do I go into energy, right? That's a big thing. Energy and environment, I had that opportunity. And then I had that opportunity to be in the biofield, within the same lab even. So then I decided that, it's been my passion to kind of biology, chemistry, physics, engineering, all of these have been something that I want to be involved in. So then I got into this the sensing world, the biosensing world. And, I kind of fell in love with that because I got to interact with chemists, physicists, physicians, biologists, and I felt it's not limiting me.
Right.
And I guess the rest is
history.
History.
I find that really interesting that I never really thought of that field in the way that you describe it, where it intersects everything together. You really have the freedom to do whatever you want, essentially. That is really a cool thing.
Yeah, exactly. Yeah.
And I'm curious, is there any specific experiences during your undergrad, your master's, maybe even your postdoc or PhD, where you were this is a really amazing like field, especially in biosensing? I know you also do like nanofabrication as well. Was there any like specific experiences that really pushed you down that path?
Yeah, that's a good question. I think in my PhD, it was really the opportunity to interact with people outside my field. So I just give you an example. I mean, I was working, in a chemistry lab, most of the time, and then a time came and my supervisor took me to a meeting and said, well, we're ready to now look at prostate cancer samples and tissue samples. And so, I got to work with oncologists at University of Toronto affiliated hospitals and really, I received these really important samples and have the opportunity to interact beyond that,
Right.
small basement chemistry lab, and just saw that this is real. I think that was really important, that I just felt like, okay, I'm not just pipetting in the basement, but this matters. This
Right.
matters to this bigger community, and it has a path to making a difference for patients. And I think that was really inspiring,
Right.
and just kind of inspired the rest of my career. So those initial interactions with the real clinical world.
You kind of allowed that to lead you into focusing a lot more on like applying the platforms and the interfaces that you work on, as opposed to just simply focusing on the science of it. I think so.
Yeah, like the
Yeah.
translation piece.
Yeah.
I think that's really important. And a lot of times, like we always say, you do a PhD, you start knowing so much about so little. But then working on a translational piece, yes, maybe that's where, the niche of my science, but then I can plug into the niche of somebody else, and then
Right.
through that, see the translation of my work and increase its impact.
Right. Especially like I feel like in the lab so many times when there's so many setbacks, it's kind of hard to, find the courage and the motivation to keep going. So I definitely think, seeing that your work is being, used in real people, in real people's lives, and helping people must be such like a motivator, right? Especially during those kind of setbacks and challenges.
Yeah, for sure. I mean, when you look at, your progress over days and weeks, it's hard because, you're like, well, I've been working, and I hear students say this, I've been working on this for like weeks and months, and I've just been troubleshooting. It doesn't seem like I'm going forward because, I go one step forward and then one step back. But I think once you look over years, then you start seeing that
Right.
you are making a difference. You are contributing to science. People do get to know your work and appreciate your work, and people from companies and different fields, right? They contact you and they want to work with you to solve problems. I
Right.
think. Yeah, so you're right. It is challenging, but it's kind of a long game, I feel.
Right. So you got to kind of keep pushing forward
Yeah.
and, have the future vision, right?
Yeah.
Makes sense.
And just thinking about your PhD, what did you go on and do after that? I mean, what did you do from there until when you became a PI here at McMaster?
So I actually is kind of a rare case because I didn't do any postdoctoral
Oh,
training.
wow.
Which is something that's rare. It wasn't common then. It's even more rare now.
Yeah.
People have a lot of advanced training after PhD. But I was fortunate, again, because of the field I was in. I got such a well-rounded and rich experience that I felt ready to start my own thing because, I participated in basic science, translation, as well as some commercialization efforts. So then I had a well-rounded experience, gave me confidence that I could do this, and it seems like it gave confidence to people who hired me, that they thought, well,
Right.
she could do this. So I came right here,
Yeah.
and I just started.
And what year was it that you came over to McMaster?
I came in 2011.
2011.
Yeah.
Wow.
Yeah.
That's you've been here for a while then.
I've been here, since
Yeah.
in my 20s. Like,
Wow.
yeah.
Very impressive, especially because I was saying this before we were recording. Like everyone knows about you, all the labs are working with you. So I'm kind of curious then. Was that the mindset you first had coming here? Did you know you wanted to build such a close knit community where you can talk to different PIs and have so many different options in terms of projects you could possibly be running?
Yeah, I mean, I think it builds on some of my earlier experience, right, of being kind of in a space where I got the opportunity to explore and collaborate, and I wanted to build something like that here. I wanted to work on meaningful problems, and as engineers, we don't necessarily know what those meaningful health problems are. We know what the hard problems are.
Right.
We know, it's hard to detect this at this level, with this kind of accuracy. But that's not really a real world need. That's just a hard problem to solve. So we love that. But to really know what are the meaningful problems for clinicians and patients, you have to kind of build those communities. Even beyond that, right? Even forget about translational piece. Even within physical sciences or physical and health sciences, there may be, hard problems I know how to solve really well, but other problems that it would be really, really hard, I wouldn't know how to solve, but somebody else has figured it out. So it makes a lot of sense to work with people, and it's both. It's not just for me. It's not just for the outcome that I'm going to get better papers and better research. It's also a lot more enjoyable,
right.
And it's more meaningful, also personal experience, but also similarly for my students, right? You've talked to some of my students. I think it gives them a rich, the multidisciplinarity of it gives them a really rich experience.
Yeah, and just kind of, I guess, going more into the science of what you do, was that kind of vision what you brought in with you when you started your group here? Was that kind of what you led with? Is you wanted to not really focus on just the I guess the science of sensors? You want it to be as multidisciplinary as possible, or did you maybe want to start out like here in McMaster focusing on more so the bio interface side of things, I guess?
It's interesting you asked that. I can't say that I knew. I mean, I kind of got here and I thought I'm going to figure this out. I've got this. But I've done a lot of different things to get here, right? I mean, I started with thinking, well, this is my skill set, right? I'm an engineer. I could do, as you said, nanofabrication. I'm also an electrochemist. And I also know quite a bit about material science. So this is my skill set. What can I do with it? And I've done a lot of different things. So now I'm very focused on sensors, but it wasn't always like that. I've explored even building interfaces for the energy industry, right, as anti reflection coatings, and I've done the work that led to Fendex together with Tohi Didar on omniphobic surfaces and as well as sensing and different areas. And I try to be open. I mean, stay within my skill set. I don't want to be in a place where I can't contribute, but a lot of times these skill sets are versatile, right? And I'm saying, okay, this is my skill set. Where can I contribute that makes the biggest difference? So I've explored those, and it's always come back to the sensing and the multidisciplinary area and mostly clinical applications of sensing. But it's come through a lot of trial and error. Building a lot of relationships with colleagues and seeing where their interests are, where the mutual interests are, where the industry interests are, where I could get funding. So it's been a lot of inputs and a lot of trials and error.
Yeah.
You also mentioned that you had like some commercialization experience after, you got your PhD. I'm curious, where did that come from?
Yeah, so during my PhD, so I worked on this infectious disease test. So it was a rapid bacterial test that we looked at detecting bacterial messenger RNA for bacterial identification rapidly. And so this test kind of formed the foundation for a company that spun out of the lab. So I wasn't directly the person who co-founded the company,
Right.
but I was, involved with the company and some of the R&D that happened in the laboratory. So I got that kind of first exposure to commercialization throughout my PhD.
That must have been super useful. I mean, I imagine that you said it yourself, all the collection of all these different skills, especially like the business skills, is so important in labs, right? It's not only about like writing grants, but also forming those connections, not only like within the university, but if you want to collaborate with hospitals, you said there's so many clinical applications. So that knowledge must have been really useful for you.
For sure. And I mean, I think I was very lucky. I was in that environment. I had great supervisors. I'm hoping that I'm building similar environments within my lab so that my trainees could have that well-rounded experience, right?
Yeah.
Because you do need that really strong, basic science research and the hard work that goes into that. That's the foundation, that's the core, but there is more. Right. There is the translation, there is the commercialization. So to have a well-rounded experience, you kind of need the other pieces as well.
For sure.
And did you run into any challenges in particular when you were first starting out your group here? Because I imagine it must have been a very radical shift to go from just being a student to being a principal investigator. That must have been a very there was no gradualness to it.
Yeah, it was just a sudden jump. Boom.
Yeah, sometimes those are actually good because you don't know how challenging this is going to be because you're so naive.
Yeah.
And sometimes a lot of times I take PhD students straight from undergrads. I'm like, it's okay being naive is actually good, because you're just like, I'll try it, and so I had that attitude, but it wasn't easy. I remember, when we went with my department chair at the time, Harold Hagen, who really helped me build, a really strong foundation. He took me to the lab and he's like, "This is the lab," and it was like this empty space, no cabinets, no no fume hoods, nothing. So we're just gonna have to now build it. And I thought, okay, how do we do that? So, I got a lot of support, but it took a year and a half to build that lab, right? And then another year to get the equipment in and the people in. So that was challenging. I'd say, right, just getting started and being anxious. When am I going to get started? I've been here 3 years. When am I going to start being productive? And that kind of loss of productivity.
Right.
So I would say those were the really challenging times.
It just takes time to get the ball rolling,
exactly.
so to speak.
Yeah.
And kind of focusing more on research of the research you've been doing and the challenges associated with that. I know your lab works on creating materials to interact with biomolecules, even as small as the nanometer scale. So I'm curious, what are some challenges with developing materials that small, and how is that? I guess how could that be useful, especially in terms of clinical applications?
Yeah, I guess the biggest challenge in this materials nanofield is repeatability. This is the challenge of our field. We can make very cool materials and we can get amazing electron microscopy. For the longest time, this was my hobby, to get these really nice images and be so impressed by them. But I think the biggest challenge when you want to demonstrate a material once and publish a great paper, you do it and you get these beautiful pictures and you show some functionality. But when you want to make a real clinical tool that one day is going to be a product, you're going to have to make them all to be the same. and same performance. It's not a problem that hasn't been solved. It's just really challenging. I mean, if you think about having 1 billion transistors inside your computer chips, right, with every one of them being the same and being as per spec, it's possible, right? The semiconductor industry has figured it out. It's quite costly. You need all those crazy clean rooms to make them, but it is possible. So I think that's the biggest challenge of our field, to make something that can be scaled reproducibly, so that it can actually be used.
And are there any like partnerships that you've tried to create to kind of address that issue?
Yeah, so we work now, as much as we love to make our own materials, so we do that for proof of concept, but we've built partnerships with manufacturers that have figured that out, are making these materials in the clean room.
I see.
Using processes that have quality assurance built in and can generate the kind of materials we need for clinical applications.
Yeah.
So we have taken a step towards that as well.
Okay, that's really cool.
And in regard to your sort of biosensor development, do you start with the mindset of going for a specific target, whether clinically or any other application? Do you start with the target or goal in mind, or do you focus around establishing the platform and then building on top of it after?
You know what? I mean, I'd love to say we're so target-oriented and we have a pipeline. It's the dream to be really clinically integrated and, just be there immersed within, the hospitals or the clinical practice and really identify what the needs are and then go and build something that can then directly be translated. But the reality is we don't always do things that way. Sometimes, as I said, we want to work on really hard problems. So we say, what's the hardest thing, like a low abundance protein to detect in a really, really busy matrix. So we start with that. And sometimes we do have those pathways, but sometimes we do it this way and then go and shop around, go and talk to people and be like, oh, I've got this thing, and what do you think we can do with it? So I think that both are effective, right? I feel like you kind of have to do both. We do kind of target-focused development, but sometimes we develop this, and then, that's what I've been doing the past couple of weeks with one of our platforms. I tell people, we can detect this molecule, at a really, really low concentration. What do we do? And I'm fortunate because we have a lot of colleagues who are interested and can sit with me and say where we can use it.
Yeah, and that kind of leads me to thinking when you do want to maybe try to push for this to be something that is point of care or rapid or actually used in industry in some way or another, how do you choose? Do you usually tend to target quality of detection, or do you go for more cost-effective of measures? That's a very fine line to tread, where you want something that's accessible but also accurate.
Yeah, that's a really good question, because it's, I think we used to think that you need high sensitivity and high precision sensors, and that's what's going to be needed to make a clinical impact. More and more we're hearing, you want something that's rapid and accessible. It doesn't have to be perfect. And, more and more, during COVID-19, right, we saw that rapid antigen tests that are, they're inferior in terms of sensitivity and specificity, but they're accessible. They can be broadly distributed, right, for a specific application. So I think there is a move towards, kind of a, how do you say it, like taking the trade-off seriously and saying, okay, maybe if I don't have to have this, standalone instrument, but I have something handheld, it's okay that I'm going to use it more frequently and more widely, but it doesn't have, the optimum performance.
Probably because of the fact that in a pandemic you also have so many people, and then if you're doing like something like a PCR assay to test for people having a specific disease positive, it would be a lot cheaper and probably more accurate to just distribute a bunch of, or probably better for public health to just distribute these really cheap rapid detection sensors. And is that something that you've kind of oriented your lab around designing for problems with in mind?
Yeah, so that's what we do. We do, we call it like sensors for frequent monitoring. And that's a term I like to use because, you can think of things like continuous monitoring, like the continuous glucose monitor, but you can also think about, using a rapid test that you can use every day when you're having, flu-like symptoms, right? So that's frequent monitoring that you can't do PCR every day, right? Mm-hmm. So, I mean, you could, but it's the workflow is complicated and expensive. So that's where we are. We are the frequent monitoring lab with a focus on wearables. Mm-hmm. Next generation wearables going beyond glucose, what glucose monitors can't do, but also point of care diagnostics that is rapid and easy to use and inexpensive.
And are there any implications of using that, having that technology be used by physicians or nurses who may be less familiar with the technology? Because I know one of the previous PIs we had on, Dr. Hildebrandt, am I saying that right?
Yep.
He was mentioning that sometimes when he's developing the biosensors, because he also does biosensors,
right,
that he has to take into consideration that the people who are actually using that technology might not be very familiar or might not know as much about how to use that technology as him or people in his lab. So is that something that you kind of have to take into account, especially, I guess, near the end of your project when you have to make sure, this technology is useful in a clinical setting?
Yeah, I mean, that's... Dr. Hildebrand is right. That's an important consideration, right? When you say rapid diagnostics, there's no point if it's rapid, but then you need a whole team, right? To come to your house and run that test. So we try to develop systems that require as little manual interventions as possible, right? We try to remove steps like sample processing and additions and washes. And part of what we do, which I find is the coolest thing, and this is something that we could do because of people like Ying Fu Li, we get the molecules to do the work, right? Mm-hmm. We, instead of manually adding things, we have molecules who actually have the reagents on them. And instead of washing, we look at materials that are anti-fouling and we don't have to do washes. So that's kind of built in within what we do to minimize kind of the complex steps. The outcome is that it's much simpler to use, but it also gives us an opportunity to be creative,
right?
To get the molecules to do the work, so we don't have to do them.
Yeah, and that's super interesting. So, are you saying that you can kind of make it almost automated in certain parts of it? I'm curious. Could you kind of talk about a little bit of the specifics of that?
Yeah, absolutely. When I said that I started my career making infectious disease sensors in my PhD. I mean, that sensor required, a lot of steps. You had to do RNA extraction and reagent addition and a lot of washes. Then when the second generation or the following generations of bacterial sensors, infectious disease sensors that I've done here together with my collaborators, including Ying Fu Li, is one where we build in the reagent in these molecular switches like DNazymes. So that reagent addition is not something we have to do manually. And then later, through other collaborations, we have looked at surfaces that are anti-fouling, so we don't need to do thorough washes to get rid of unwanted materials. You can operate on those surfaces, and interference is going to be minimized. So to go back to your question, so again, it goes back to materials, different kinds of materials working together so that we can have optimal sensors that are easy to use.
And I kind of wondering, like, I know one of the techniques you use is the in situ liquid transmission electron microscopy. Does that play a role in that process?
So that's something that I was very excited about, coming here, because we have one of the world's most advanced electron microscopy facilities at McMaster. And in fact, that's one of the things that I knew the most about McMaster before coming here. And so I really wanted to build up that capability so we can do wet kind of work, like not just look at metals and steel and semiconductors, but look at dynamic biomolecules. So that's the work I started here. But it's one that I didn't really continue. It's a capability that exists here now, but we don't focus on it so much. It's quite a challenging project, and most of the funding that I've received over the years has been on the sensor side. Didn't leave a lot of resources for doing that kind of more fundamental imaging, so I've kind of left that behind for now.
Are there any like potential projects you're thinking of that could be using that technique, or
there are people who are great electron microscopists here at McMaster, like my colleague Catherine Granfield. So we're fortunate enough to have her. So, when we do need that kind of more advanced electron microscopy, and especially in liquid, we normally go to her now, and she normally, always says yes and helps us with all of that. So I've kind of, let her
handle that, yeah.
And just talking a little bit about the wearable sensors that you mentioned earlier, are there any potential implications of the research that you're doing in that sort of sphere? Do you foresee maybe technologies that you're working on right now to actually become something that goes to market in the next couple of years, maybe?
Yeah, I'm really glad that's my favorite. I'm really glad you brought that up, because that's kind of my focus. Like, I wake up in the morning thinking about that, and I go to bed thinking about that. I wake up in the middle of the night thinking about that. So we actually started a company called Optic Health to commercialize the next generation wearables for personalized health. And we have two products based on this technology. One is focused on monitoring cardiac troponin, so it's a biomarker for cardiovascular health, and we want to monitor patients after surgery that might have a heart attack, but we can detect that early. The second product is on an insulin sensor, so insulin, not glucose, just to be able to detect insulin pump failure at an early time. So that's what we're working towards, to get this technology to the hands of people in the next few years.
That sounds really interesting and probably very, very useful for I can imagine a lot of people would actually benefit from this kind of thing. I mean, I know a lot of diabetics myself in my family, and some of them do use insulin pumps. That is a fear that they have sometimes. They have to look out for that kind of thing.
Yeah, and then are you currently doing any like trials for that? Is that like something, like, how does that work actually? I've never kind of understood the process.
Yeah, so I'm trying to, I mean, for me too, it's kind of interesting. Every day it's a new learning experience with that. But so normally what we've been doing, we do in vitro validation. So on the lab bench, we have a prototype, we validate that. And then what we've been doing, then we go to in vivo work, and we're looking at different animal models to validate those, which is where we are now.
Right.
The next phase is to do the first in human or pilot human trials, which is something we see ourselves doing in 2026, both for troponin and insulin. We have that mostly finalized through other trials that are happening. But I mean, there's quite a long journey
Yeah.
ahead of us, because
I'm sure.
there's a regulatory process
Right.
that we have to go, both for first in human trials to get kind of a test authorization, but also for selling these, right? You have to go through Health Canada or FDA.
And just kind of wrapping everything up, what would you point to, I guess, as of recent time, as something you're particularly proud of that you or your research group have both done together?
You know, I think the best thing about this job is the opportunity to work with students. I think I'm the most proud of their successes. And
Right.
I always say, like, my students, my trainees, whatever it is that they want to do, I think they're quite successful. They whether they go to industry or academia. So that's what
Right.
will stay as kind of my impact beyond kind of this small
Right.
research group, and that's what I'm the most proud of.
It must be so gratifying to kind of see your students kind of take up your torch and go into so many different paths, right? I mean, that's kind of what science is all about, in my opinion. Doing all this work is ultimately for the betterment of humanity, and kind of seeing that, your work is being, passed on to the next generation, and then from the generation after that, and then the generation after that is like probably like the best feeling, right?
100%. And I said, like, you don't see the impact over weeks,
Right.
Yeah.
right? It's over
You just
years.
randomly check up on someone one day and they're like, oh, this person.
100%, yeah.
Well, thank you so much, Dr. Soleymani, for coming down here and talking about your story and your research. It's been great having you on and getting to meet you finally. And thank you to everyone listening, and we'll see you next time on Office Hours Unplugged. Thank you, Dr. Soleymani.
Thank you.
Thank you.